let's start talking about the hippocampus, facts and myths. So, the hippocampus, what is it? Where is it found in the brain? The hippocampus is a very, from the evolutionary point of view, a very old structure, and it's quite a small structure in the human brain. It only occupies about 2% of the brain. It's located on the medial side of the hemisphere, so meaning the part of the hemisphere that you'd find that you wouldn't be able to see on the surface when you look at an MR image, and it's located in the temporal lobe, okay? And it is a long and thin, sort of like sausage-like structure, which you can see highlighted here in red in this animated GIF. Now, as I mentioned, it is a really, from the evolutionary point of view, a really old brain structure, and also its name dates very far back in time. So the name hippocampus was first used by the anatomist Arancius in 1564, okay? And what he said was that macroanatomically, the hippocampus, this part of the brain, reminded him of this little sea animal, so it's an upright swimming fish. And he also said, well, I'm not sure if it reminds me of this animal here on the left, or if it reminds me of this animal on the right, which is a little white silkworm. I think for us nowadays, thank God, he decided to go for this species here on the left, and that's why it's called the hippocampus, which is the Greek word that is used to designate this small upright swimming fish, okay? So you see, anatomists, we've got a lot of imagination sometimes. Now, we got thinking about what other things does this structure here remind you of? And if we take it and we just rotate it, okay, by 180 degrees, and we like turn it upside down, and here you've got another view. Now, what would this remind you of? Well, this is again another anatomist with a lot of imagination. In this case, it was Golgi, who was one of the really important names in neuroanatomy, because he developed the staining that enables visualization of entire cells. He thought it reminded him of a paw-like structure, right? So on what you've got here on the right is a photograph of a paw of a large lion, and it doesn't get much bigger than lions, and that's why he called this rostral part of the hippocampus here, he called it pes hippocampi, so Latin for foot of the hippocampus. Or we also talk about, because there are some small sulci here, digitationes hippocampi, these are the fingers of the paw of the hippocampus, okay? So as I said, a lot of fantasy in the way that anatomists have given this structure its name. Then we come to the term cornu ammonis, okay? It was again described or named a long time ago, namely in 1742. And here we had a French anatomist, and he said that the hippocampus, when viewed from above, it reminded him of representations of either the Greek god Amun-Ra, or of the latter in history appearing figure of the Greek god, the combination of the god Zeus and Amun-Ra. Now, from this picture here that the Gallagher made, it might be a bit difficult for you to see the similarity, but if I show you this image here, which is a plastic model of what this looks like. So what we're looking at here is we're looking at a human brain from above, and most of the frontal lobe, most of the parietal lobe, and a lot of the temporal lobe. So all of this part here has been removed, and you can see the ventricles, which are these structures here, and then you can see the hippocampus, which bulges into the ventricle. And I think looking at this figure here, well, it would maybe remind one of the god Amun-Ra, okay? So that's how the cornu ammonis got its name. And up to here, it's amusing that anatomists use their imagination and have all of these names. But sometimes anatomists can be irritating because the problem is that they're not always consistent in the way they use names, and sometimes this is historically happens that two anatomists had been working on the same structure and each one gives this structure its name, and then of course they can't decide on, you know, who is the father or the mother of the name. So the structure gets both names. So the problem is that then in the literature, you might read the words hippocampus or hippocampal formation or hippocampus proper or hippocampal complex. And you might wonder if these structures or if these designations are synonymous or not, and if they can be just freely used. And the answer is no, they're not synonymous. And unfortunately, there is no consensus about what structures are encompassed by these names. So I will try and give you an overview. This is part of what today's talk is about, okay? So that you learn the difference between some of these names. And just the same as you've got differences here between, you know, hippocampus, hippocampus proper, we also have then differences when we're talking about the cornu ammonis, the dentate gyrus, or the subiculum. When I've used the same name, so for example, here for the dentate gyrus or the gyrus dentatus, these two terms, they are synonymous. It's just that one is the Latin designation and the other one would be the English designation. In the moment in which I've used different colors, it means that these are not synonymous designations, okay, which, as I said, can be confusing. And let me just show you an example. So when we use the word hippocampus or hippocampus proper, some anatomists mean the part that is highlighted by this blue surface, okay? Whereas other anatomists are designating this here. You see, the difference between these two images is that this part here is either part of the hippocampus proper or not. Some other anatomists think that hippocampus proper encompasses all of this here, okay? Other anatomists, and this is the view that personally I endorse, think that this surface here that is surrounded by the red line is what we call the hippocampal formation or hippocampal complex. Some authors would argue that the entorhinal cortex is also part of the hippocampal formation, okay? I don't belong to that school. I suppose this is just a question of tastes, and it's a question of, at the beginning of your article, really then defining and specifying what it is that you include in the term hippocampal formation or not. So then those of us that think that here the entorhinal cortex isn't part of it, we would say that the hippocampal formation is composed of the hippocampus proper, or just for simplification, the hippocampus and the subicular complex, okay? Some authors also simplify, and instead of talking about subicular complex, they talk about subiculum. Now this is a problem because within the subicular complex, there is an area, a cytoarchitectonically, receptorarchitectonically, and connectionally distinct area, which is actually called subiculum, right? And it only occupies this part of the subicular complex. So again, if you are simplifying, simplification is sometimes good, but simplification can lead to misunderstandings, and misunderstandings can ultimately result in misconceptions. And this is what leads to us not really advancing in science. Anka.
So, I have a question. So, would you say that from this image you could separate the speculum from the vicinity based on cytoarchitecture?
So from this image here, repeat please. Yes, so what Anka wants to know is from this image here, I can separate the subicular complex from this part of the cortex here, or from this, if I can establish these borders, right?
No, the subiculum proper.
Oh, the subiculum
yeah.
proper. Now, from this image here, at the resolution at which I am showing it right now, I could say vaguely, yes, if I wanted to be sure about it, I wouldn't do that at this resolution, I would zoom in. And that is actually what I did to create this slide. And then I asked Olga to confirm my borders. And, you know,
Double checked.
yes, exactly. Okay, so not, yes. To be exactly sure about where I put the border down here, that was definitely something that I had to do at a higher resolution. Okay? Anyway, today I'm not going to talk about the subicular complex anymore, so not much more, so it doesn't really matter. Anyway, as I said, during my talk, the convention that I will use is that the hippocampal formation is composed of the hippocampus proper, which I might, or most probably will always just shorten into hippocampus, and the subicular complex, okay? Now, if we now look at a view of the macro anatomy of the hippocampus. So again, we've got a human hemisphere here. You've got to imagine that what we're looking at is a right hemisphere, and that the medial wall, so the part of the hemisphere where both hemispheres are basically contact each other, that part of the hemisphere would be facing in towards this room here, or towards you and your computer screen, and that the lateral surface of the hemisphere would be behind the wall. Okay, that's what we've got here. And to make the structures that we want to see more visible, that the frontal part of the hippocampus, where we had the foot of the hippocampus, the pes hippocampi, it's been slightly cut away. Okay, and here it is possible to see the two main structures that form the hippocampus proper. We've got the so-called cornu ammonis and the fascia dentata, and this is what anatomists describe as the two interleaving Cs, okay? And when we look along here at the surface, you can see little lumps. This is again where anatomists used a lot of imagination, and they say, oh, this reminds me of when you look at teeth, right? If you take your you look at your teeth from above at your molars, that's what this reminds me of. So that's why they called this part here of the hippocampus, the one that we can see from the outside, macro-anatomically, they called it the margo denticulatus. Okay, so denticulatus is the toothed margin in Latin. That's what margo denticulatus means. And that's what is then nowadays called the dentate gyrus. Okay, so the fascia dentata is what you would see in a cross section, and the dentate gyrus is what you would see macro anatomically from the surface. And just for you to see another view here, where we also have the uncus part of the brain, there's this example here, okay? And just the third definition of hippocampal parts, macroanatomically, the hippocampus is divided into three portions, so which are approximately a third in size each. The most rostral portion of the hippocampus, where the pes hippocampi are located, is what we call the hippocampal head. Then the central third is what we call the hippocampal body, and the caudal third is what we call the hippocampal tail. And what you see here is a coronal section through a human hippocampus, through the part of the human hippocampus where we would have the body of the human hippocampus. And I have highlighted the cornu ammonis and the fascia dentata in blue and in red, respectively, okay? And these are, as I said, cytoarchitectonically identifiable structures, right? If we then merge the CA4 region, so that the cornu ammonis can be subdivided into CA1, CA2, CA3, and CA4 regions based on differences in cell body packing, based on differences in the distribution of receptors for classical neurotransmitters, based in connectivity patterns, okay? And if we merge the CA4 region with the fascia dentata, then we get what is called the dentate gyrus, which is this structure that we can see macroanatomically, okay? Now, in a human brain, I mentioned earlier on that the hippocampus is located on the medial surface of the temporal lobe. So here we've got a coronal section through a human brain, which has been stained for cell bodies. And if you remember, and I've highlighted the hippocampus on both, you know, on each hemisphere with these black squares. And if you remember one of the talks that I gave in earlier HIBALL workshops, you know that the cell bodies aren't equally distributed throughout the brain, that we've got parts where the cell bodies are basically localized, and when this part is found on the surface of the hemisphere, we talk about the cortex, and when these cell bodies are located inside of the brain, we talk about subcortical nuclei, okay? And everything else that is in here is the white matter. So up to now, I've told you facts about the hippocampus, things that you can read in the literature and you may believe. Now, the problem is that sometimes things are published and they're wrong because we editors and we reviewers didn't pay attention, right? And incorrect information gets published. And the problem is that this incorrect information, it gets cited because people read it and they say, well, if it's been published, it must be true, right? So then you read that the hippocampus is a subcortical structure, which it isn't. And I've just got a couple of examples here, you know, where you can see cortical, and then in brackets, frontal, temporal, parietal, and then subcortical, and you've got the hippocampus with the amygdala together as a subcortical structure. And this is a very new article. This one was published in 2024. This goes back a long time. I didn't go back, you know, to the first time that this was published because I don't know when it was, and I didn't really want to waste time on that. But you know something, this has even been published in Science. And everybody thinks, well, you know, if it's been published in Science, it must be right, because Science is such an important journal, okay? So here in this Science article, they didn't write in the text that the hippocampus is a subcortical structure, but this entire article was about changes in subcortical structures. And in their table, they have a list of the subcortical structures that they analyzed, and you can see the hippocampus here, okay? So if you read anything anywhere that the hippocampus is a subcortical structure, it's wrong, okay? I always try and think, where did this come from? And I'd say it's basically just not enough anatomical knowledge of the people that wrote the article. I have seen it most often in the rat literature. And I must say, when you look here, this is a coronal section through a rat brain, and it shows you an image of the dorsal portion of the rat hippocampus, which is equivalent of the anterior portion of the hippocampus in the human brain. Here, it does look, you know, down here you've got basal ganglia and you've got thalamus, which are subcortical structures. And you've got the hippocampus here underneath the corpus callosum. It doesn't seem to have a connection with the cortex. So maybe this is where the mistake comes from. Mind you, if the people just took the trouble and looked at a section, further caudally in the rat brain, where we have the so-called ventral hippocampus in the rodent brain, which is equivalent of the posterior hippocampus in the human brain, then you would see that the hippocampus definitely has a connection with the cortex, and it just confirms that the hippocampus is a cortical structure. Okay, right. Now this is another slide that I've shown in previous talks, and basically I just want to remind you that the cortex is built up in a laminar pattern, and we've got areas with different numbers of layers. Okay, but all of this that you see here, from this point here, which would indicate the surface of the brain, to this point down here, which would indicate the interface between in this case, layer 6 and the white matter, all of this is the cortical gray matter. Okay. I'd also like to remind you that we've got different types of cortices depending on the number of layers that can be identified within them during ontogeny, and we've got what Brodmann called the homogenetic cortex and the heterogenetic cortex, and that is what we nowadays know as isocortex and allocortex, okay, respectively. And again, just remember, all of these layers constitute the gray matter. Now, we've got isocortex, we've got allocortex, and between the isocortex and allocortex, we've got another type of cortex, which is a transition between the two of them, and it's called mesocortex. And these are types of cortex that can be identified during ontogeny, right? So in the embryonic brain. And if we want to look also at types of cortex that can be identified based on the analysis of different animal species, then we've got the phylogenetic classification of cortical types. And here we've got paleocortex, which is by far the oldest one, okay? Then we've got archicortex. These two types of phylogenetic cortical types belong to the allocortex. Then we'd have periallocortex and proisocortex. These both types of cortex belong to the mesocortex. And then we have the neocortex, which is synonymous of isocortex. So these two terms here, you can pick and mix as you want, because with them we're representing exactly the same portion of cortex. Okay. Now, why I'm going into all of this detail is because the hippocampus belongs to the archicortex. Okay. And I talked earlier on about the fascia dentata, and I talked about the cornu ammonis. Okay, so on the left of this slide, you've got a schematic representation of the layers that can be identified within the fascia dentata, and on the right, those that can be identified in the cornu ammonis. Now, per definition, the archicortex has 3 layers. And in the fascia dentata, we've got the so-called molecular layer, we've got the principal cellular layer, and then we've got the multiform layer. The principal cellular layer contains the cell bodies of the so-called main neuronal type in the fascia dentata, and these are the granular cells. That's why this layer is called the granular layer, okay? And the output of these cells, so the axons of these cells, they build the so-called mossy fibers. Now we know that within the molecular layer, we could define 2 sublayers if we want to, based on differences in connectivity, and some anatomists just talk about the molecular layer, but if you're interested in connectivity, then you should definitely talk about the inner and the outer molecular layers, okay? Because the outer molecular layer is the one that receives input from the entorhinal cortex, and basically the inner molecular layer is the one that receives information from the hippocampus from the upper hemisphere, okay? This is very simply put the main difference between these two parts of the molecular layer. In the cornu ammonis, we again start with only 3 layers. So we've got the outer plexiform layer. It is the outer plexiform layer because this thick line here in the graph represents the pial surface, okay? And this thin line down here represents the border between the gray matter and the white matter, okay? So we've got the outer plexiform layer, we've got a principal cellular layer, which in the case of the cornu ammonis is composed of pyramidal cells. That's why this is called the pyramidal layer. And then we've got the inner plexiform layer. Both the outer plexiform and the inner plexiform layers can also be subdivided in the cornu ammonis based on differences in connectivity and also based on differences of the types of cells that they occupy, right? So in the outer plexiform layer, we've got from the pyramidal layer moving outwards towards the brain surface. We've got the lucidum layer, which is only present in the CA3 region. That's why I got a little asterisk here. Then we've got the radiatum layer, the lacunosum, and the molecular layers. Some anatomists talk about radiatum, lacunosum, and molecular layers as 3 separate sublayers. Most commonly, the lacunosum and the molecular layers are merged together to form and are designated the lacunosum molecular layer, okay? And sometimes, just depending again on what your scientific question is, people will just talk about the outer plexiform layer. In the inner plexiform layer, we've got the Oriens layer, and we've got the alveus, which actually only contains the axons of the pyramidal cells. So it's by some authors, it's also designated as the hippocampal white matter, okay? So I've shown you these different layers now as a schematic representation. What would they look like if we had a look at the coronal section that I showed you earlier on? Well, we would have here this dark band in here that I'm going to highlight in red. We've got the hippocampal fissure first. Now, the very first talk that I gave at the very first HIBALL meeting, I told you that the brain surface isn't smooth, that it's convoluted, and that the convolutions are called gyri, and that the boundaries between gyri are called sulci, unless they're very, very deep, and then we would call them fissure. So here you've got basically a sulcus, which is not in the least deep and prominent, right? I mean, it's prominent because you don't see it. And it's called the hippocampal fissure. It's called the hippocampal fissure because when the hippocampus was born, it wasn't convoluted. I'll go into a bit more detail about that later on, okay? But here's where anatomists, again, aren't always 100%, yes, well, consequent in their naming. Let's just put it that way. Right, so in red, I have highlighted the granular layer of the fascia dentata, so the cell bodies of the granule cells which form the fascia dentata. Then towards the brain surface, we have the molecular layer, and inwards, we have the multiform layer. The blue surface highlights the pyramidal layer, which contains the cell bodies of the main cell type in the CA region. And as I said, it can be divided into CA1, 2, and 3 based on differences in different criteria. We then have, when you move from the pyramidal layer deepwards, right, towards the white matter, we've got the Oriens layer and the alveus layer. And when we move from the pyramidal layer towards the brain surface, we've got the radiatum layer and the lacunosum molecular layer, okay? And just in the CA3 region, we've got the lucidum layer, okay? So all of these layers constitute the so-called gray matter of the hippocampus. And here again, I come to another myth, another thing that you will find published in the literature, where the authors describe that the hippocampal gray matter does not include the radiatum, lacunosum, or molecular layers, right? And according to this definition, only the lucidum layer, the pyramidal layer, and the Oriens layer would constitute the hippocampal gray matter. I don't understand the logic of this end of the story. The authors underpin the opposite. They underpin their statement with a reference by Duvernois. Actually, what Duvernois said was that the cornu ammonis made it be divided into 6 layers: the alveus, stratum oriens, pyramidale, radiatum, lacunosum, and moleculare. And when Duvernois says may be divided, they don't mean include these layers if you want and exclude them if you want. What they mean is you can either identify 3 layers, the outer plexiform, the inner plexiform, and the cellular one, if you want to have the lowest level of granularity, or identify up to 6 layers if you want to go into the highest level of granularity, okay? So as I said, again, this is published, it's not correct. That hippocampal gray matter is limited to just this part of the cortex that is highlighted in gray. Now, I mentioned that the radiatum and lacunosum molecular layers basically, I didn't mention it maybe, but they basically only contain the dendrites of the pyramidal cells, but they also contain other types of cells, okay? And the different types of cells that we can find within the cornu ammonis, an example of the number of cells that you can see dates back as far back as 1893. I mean, we're going back into the 19th century now, okay? So we've known for a long time that there are other cells in these layers and not just dendrites. And these are drawings that were made by Ramon y Cajal. And on the left, there are drawings of the pyramidal cells, so the main cell type in the cornu ammonis. And here on the right, we've got the different layers that I just described earlier on. And then he's drawn different kinds of neurons that you can find in here. And he has placed them in the layers in which they can be found, okay? And most of the neurons that are drawn here are actually so-called interneurons, so they are neurons that produce the neurotransmitter GABA. And just in a slightly later publication, he repeated these drawings of the cornu ammonis, only put them into one image, and he also provided drawings of the different cell types that we've got in the fascia dentata. Again, here we've got the granule layer, that would be the molecular layer, and down here we've got the multiform layer. Now, I talk about Ramon y Cajal, and Patricia will know who I'm talking about, but maybe not everybody knows about it, because some of you might know about Cajal, might know the name, just not Ramon y Cajal, which was his full name, but it's sometimes shortened down to Cajal, and maybe you pronounce it in a different way. So just for those of you who weren't familiar with the way that I pronounce it, or maybe those of you who didn't know who Cajal is, he was a Spanish anatomist who basically postulated the neuron theory. He was the one who discovered that neurons are actually separate entities in the brain. You've got to imagine that every neuron, every glial cell in the brain, it's like an island, right? So you've got an archipelago of islands. They're very, very close to each other. So you can see the other island when you're standing on your island, but they don't touch. There's a bit of sea between each one of them. And the thing is that this is the view that Cajal had, and he demonstrated it basically with this image here and in this publication here. This is an opposition to the view that most anatomists of the time had. They actually thought that the brain shouldn't be described as an archipelago of islands, but it should be described as a continent with different countries in it. So the brain would be like Europe, or call it the United States of America, and that every country in Europe, or every state in America, or every country in the northern hemisphere of the American continent, that each one of those would be a neuron, and that they really would be physically connected with each other. Okay, so these are the two views that existed in neuroanatomy at the time. And as I said, Cajal didn't just postulate that each neuron was a distinct entity, but he also provided the proof that this happened. And this, among other things, was the reason that he was given the Nobel Prize in physiology or medicine in 1906. And not just that, but it also gave him the title of father of modern neuroscience. And although he postulated a lot of these things in the 19th and the early 20th century, so more than 100 years ago, the advances that have been brought to us with neuroscience have only served to confirm the theories that Ramón y Cajal postulated. Actually, none of the advances has served to say, do you know something? Cajal was a genius concerning this matter, but he hadn't a clue what he was talking about concerning this other matter here, right? So everything that he postulated up to now has been confirmed with our advances. Now, you might think that I'd finished with nomenclature issues, but no, there's another one that I've got here. I've got to go into a bit more detail to be able to explain my following slide, and that is that within the dentate gyrus, right? I told you that the dentate gyrus is built, when we merge the fascia dentata and the CA4 region. The thing is, if within the dentate gyrus we identify the molecular layer of the fascia dentata, and we identify the granular layer of the fascia dentata, and then we merge the multiform layer of the fascia dentata together with the CA4 region, then we get a structure which is called the hilus. Okay, right. I just wanted to mention this name because it appears in this slide here. Now this shows results of a much more recent study than that of Cajal, where he already identified a load of different types of neurons in the hippocampus. This study here has basically, it's a meta-analysis, and we now know of at least 131 different types of neurons in the hippocampus. 21 of these 131 are glutamatergic neurons, so glutamate, they produce the neurotransmitter glutamate, they are excitatory neurons, and 110 of these different types of neurons are GABAergic. So they produce the neurotransmitter GABA, inhibitory, so they are inhibitory. And I'm not going to go into any more detail, but these 131 different cells have different laminar distribution patterns within the cornu ammonis, within the dentate gyrus. If you want to take the trouble and add up all the numbers here, you'll get more than 131. That's not because I can't do my math. It's because some cell types appear in different layers or in different regions. So that's why then they count basically twice, okay? Right, so you must be getting a bit fed up of seeing this slide. I just wanted to put it in the context of the next slide. So in the brain, it's not just that we've got layers, but within these layers, cells have a very specific orientation, okay? And this was already described, for example, by Brodmann. Brodmann here, I'm going to highlight it in color because it's too big, too small to see. Brodmann defined the so-called outer main layer, which is what we nowadays call the supragranular layer, the inner main layer, the infragranular layer, and he defined these layers based on their relative position to the brain surface, so to the pile surface, okay? So that means that in the brain, we can identify a so-called outer cortical surface and an inner cortical surface, okay? And relative to this outer and inner cortical surface, we've got our pyramidal neurons, where we know we've got the cell body, we've got the so-called apical dendrite, which is located between the cell body and the pile surface, the outer contour of the brain, and then we've got the basal dendrites, and we've got the axon, which are located closer to the white matter, to the inner cortical surface, okay? And this is something that Cajal also drew in his, drawings of the hippocampus. We've got the cell body of a pyramidal neuron. Here we've got the apical dendrite. Here we've got the basal dendrites. Here we've got the axons going out. In this part of the hippocampus here, the axon goes out here, but it has a side branch, a so-called axon collateral. I mean, this is such an unbelievable drawing. It's not just, I think it's pretty, but it is not just, maybe nobody else thinks it's pretty. I think it's pretty, but it also contains so much information, right? Because in this drawing, Cajal didn't just put, the different cells and differences in their shape and size, but he also drew these little arrows. And that is because Cajal thought that the shape of a neuron could provide information on how information flowed. And this theory of his, because he was not able to prove it in his time, because they didn't have in that time the methods, the necessary methods to be able to prove this hypothesis. This was a hypothesis that was proven right later on. Okay, we know nowadays that these arrows which he drew are the way that information flows in the brain. So, I mean, he could draw these things here because he had the Golgi stain, which had all of these things. But I mean, how do we know that then this is the brain surface? Why don't we just say that this is the brain surface? This is because I told you earlier on that hippocampus, it doesn't start life as this convoluted structure, these two interleaving C's, okay? Actually, the hippocampus starts life as a straight piece of cortex. And these schematic representations here show you the view of the hippocampus in a rabbit at 15 days of gestation, through 16, 19, right up to 25 days of gestation. And if you just draw the outer cortical surface and the inner cortical surface on each one of these lines, it becomes obvious that this dashed line here, which indicates the hippocampal fissure, is what was originally brain surface, outer contour of the brain, okay? So in this framework, it's unfortunate that the convention for hip unfold is such that the inner cortical surface is not the border between the cortex and the white matter, but it is the border between the pyramidal and the radiatum lacunoso molecular layer. And I say that it's unfortunate because you see, hip unfold sees inner cortical surfaces like this. The anatomist sees them like this, okay? So it would be easy enough if you just say, okay, I've defined these lines according to the cortical curvature. Then you can talk about inner and outer curvature without causing any kind of well, possible misunderstandings. And that is because you see, when the anatomist, here, I don't know if you can see it well, but I'll point them out. So in this view here, I have made two little neurons, okay? So we've got the neuron with the cell body, with its basal dendrites and with its apical dendrites. This is the way that neurons are located in the brain, okay? Now the problem is, if we read what's written here, that the inner contour of the hippocampus is continuous with the neocortical plial surface, and that the outer contour of the hippocampus is continuous with the neocortical white matter surface, what the anatomist sees is not this here. It sees, oops, I've got somehow some sort of a crossing here, and my neurons here in the hippocampus, they're basically doing a handstand, right? They're upside down if you compare this one and this one here. And my neurons, at least, they started to do a bit of a shortcut when I read this. So yeah, that's why I think it's unfortunate. Concerning this neocortex, this brings me to something else that's been published, and which is not correct either. And this is, you know, quite an important faux pas. And this one here, you know, inner outer contours, okay, unfortunate. This here is something that's comparable in magnitude to the fact, you know, to the description that the hippocampus is a subcortical structure. So the authors write that the hippocampus is a folded component of archicortex that is continuous with the neocortex. And again, they use the Duvernois and they use the Nivenhuis to underpin this statement. Now, the Duvernois and the Nivenhuis, they're both really nice textbooks, but they're only textbooks. They present a highly simplified view of things, okay? And I must confess that I didn't believe that the Duvernois had written this, right? Because reading that the hippocampus is continuous with the neocortex was something absolutely new for me. But I went back to the Duvernois, and because I've got it as a PDF file, I could just do a search. This was really, really easy, okay, for me. The Duvernois mentions the word isocortex or neocortex 5 times. And of the 5 times that it's mentioned in the entire book, only 2 of the citations are relevant, and that's what I've got here. So what the Duvernois says is that the limbic lobe is usually subdivided into the allocortex, which includes the hippocampus, fantastic, blah, 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 and the periallocortex, which is made up of the transitional cortex between the allocortex and the isocortex. So what Duvernois is telling us here is that the allocortex, to which the hippocampus belongs, is not continuous with the isocortex. Duvernois is telling us that the periallocortex, which is a type of mesocortex, is located between the hippocampus and the neocortex. Okay. Duvernois also says that direct connections seem to exist between the neocortex and the hippocampus. This doesn't mean that these two areas are touching each other. This means it's connections in the term of connectivity of axons from one area that are going to another area. So imagine it as you've got your islands and there are airplanes going between the islands, right? It's not saying that the two islands are touching each other. It means that there is an airplane connecting the two islands with each other. This is what Duvernois says. What does Nieuwenhuis say? Well, do you remember I showed you this slide earlier on with the different types of cortex? Well, it's highly reminiscent of this figure that Nieuwenhuis shows, right? Where we've got the hippocampal formation in pink, and then we've got limbic or parahippocampal areas in gray, and we've got these other paralimbic or proneocortical areas in the stripy view. Okay, now you must know that Nieuwenhuis belongs to the scientists that believe that the subicular complex is part of the so-called hippocampal formation, right? So he thinks that dentate gyrus and cornu ammonis are hippocampus proper. He talks about subiculum here because he has simplified the subicular complex and just into subiculum. It's not the cytoarchitectonic area subiculum, it's the subicular complex. And then we've got the indusium griseum, which is it's a very, very primitive part of the hippocampus that's just visible as a dark stripe all along the corpus callosum. And belongs to the so-called supracommissural hippocampus. Now I have, again, I'm using colors to label my different cortical types. So dentate gyrus, cornu ammonis, yellow, because they're both archicortex. The word subiculum, I have made it yellow and blue because some parts of the subiculum are archicortex and other parts of the subiculum are periallocortex. I'll go into a bit more detail about that in a second. Then we've got the limbic or parahippocampal areas, and here we've got area 28, which would be the entorhinal cortex, and is periallocortex. It is in direct contact. You know, if you consider the hippocampal formation, it's in direct contact with the hippocampal formation, but the area 28 is periallocortex. Then we've got area 29, which is also periallocortex. We've got area 30, which is neocortex, so isocortex, but it doesn't share a common border with the hippocampus, right? We've got area 25, which is located here at the front. So if you want to include the indusium griseum in your definition of hippocampus, it abuts area 25. It's periallocortex. And then here at the back, we've got THTF. This is interesting. Nieuwenhuis is mixing nomenclatures because these numbers are nomenclatures which are used by Brodmann. And the areas THTF are more areas that are used by von Economo and Koskinas, and also names that are more commonly used nowadays in monkey literature than in human literature. But then again, here we've got area TH, which is in direct contact with the hippocampus, and that is pro-isocortex. And we've got area 35, which is periallocortex. And then we've got these areas 36, 23, 32, which are neocortex, but they don't touch the hippocampus. And well, you might think up here, area 23 does touch the indusium griseum, okay? But the thing is, we know that nowadays that this drawing that Brodmann made, and this is the one that Niebenhuis based his drawing on, we know that the retrosplenial cortex, that's this area here in red, doesn't just finish here, it moves much further forward, and that area 33 moves much further back, and that actually these two parts of the brain, they share a common border. So actually, area 23 isn't touching the indusium griseum. Area 23 is separated from the indusium griseum by areas 29 and 30. Okay, so 29 being part of the mesocortex.
I have one question about this definition of areas. I would have suspected volumes. So when you speak of areas, do you refer to this certain slide of the brain?
Well, so there's a question just now about me talking about areas and not about volumes. When we talk about areas, I don't mean area as in a flat surface. I basically mean what you would call a region of interest in your PET image, where we've got the thickness of the cortex over a certain area of the brain surface. Okay, it's just a cytoarchitectonic area. It's a volume in your PET. Okay.
So the naming is then consistent over several slices?
Yes, yes, that the naming would be definitely over. If you sectioned, if you sectioned your brain in coronal sections here, you'd probably have like 20, 30 with which would have an area which you could cytoarchitectonically identify as whatever, 32 or whatever, okay? Right. This here it shows an image that Olga created, really in unbelievable detail. She delineated each one of the parts she delineated each one of the architectonically distinct regions in which the hippocampus proper can be subdivided. So hippocampus proper, we've got the fascia dentata. Then we've got the cornu ammonis region with its CA1, 2, and 3 areas. And then we've got the areas of the subicular complex, okay? And these are, it's a sequence of coronal sections. This one here on the top left would be the most rostral section. Then you'd have to move like this, like this, like this through the brain. And this one here would be the most caudal section, okay? And next to these different cytoarchitectonic entities into which the subicular complex can be subdivided. I've written to which category they belong. So only prosubiculum and subiculum belong to the archicortex, whereas the presubiculum, the parasubiculum, and the transubiculum, they belong to the periallocortex. Okay. Now, I probably lost some of you now because there were too many names and too many colors. So let me try and get back to my original question, which is what cortical type is directly adjacent to the hippocampus? And there is no easy answer to this because it depends on what part of the hippocampus you're looking at. If you're looking at the rostral portion of the hippocampus, where we have entorhinal cortex and we have area 35, or if you're looking more at the more caudal portion of the hippocampus, where we no longer have the entorhinal cortex, but we do have the transubiculum, and then we have areas 35 and 29, okay? It also depends on your view of what areas should be included in the term hippocampus proper or hippocampal complex. So if you take the more conservative view, where you say that only the fascia dentata and the cornu ammonis build the hippocampus proper, and you think that the entorhinal cortex is not part of the hippocampal formation, then you would have to say that in the rostral portion, the hippocampus is adjacent with either archicortex, because it would be adjacent with the prosubiculum, right? Or if you're talking about hippocampal formation, it would be adjacent with the periallocortex, because it would share a border with the entorhinal cortex, okay? Caudally, you would have to again say that the hippocampus proper shares a border with the archicortex, and that the hippocampal formation shares, it would, I'm sorry, I should have changed this. At the back here, we don't have entorhinal cortex. It would share a border not with the periallocortex, but with the pro-isocortex here, okay? Because the hippocampal formation would go down to here. So rostrally, hippocampal formation would abut periallocortex, and caudally, hippocampal formation would abut pro-isocortex and not periallocortex, as I've written in this slide here, okay? If you don't take this conservative view, but you have the more inclusive view, then you would say that the hippocampus proper would abut periallocortex rostrally and pro-isocortex caudally, because you've included your subiculum here, and you would definitely say that the hippocampus proper would form the pro-isocortex. I'm almost finished. I want to go one more slide, I think. So if we go back to this reference here, because the authors have stressed that the hippocampus is a folded component of archicortex, this means they're taking the conservative view that the hippocampus is only composed of cornu ammonis and of fascia dentata. So then they can't say that it abuts the neocortex, okay? They should have written that it abuts the archicortex. Hippocampal regions also differ in their receptor architecture, and because I'm over my time and Susanna's starting to get nervous, I'm sorry, I'm over my time, I shouldn't be. If you have any questions to do with neuroanatomy, feel free to drop me a line anytime you want, okay? And thank you all for your attention.